go ahead and get started. So my name is Sartin, and I'm going to talk about human evolution. And when I was asked to do this talk, I was back in April, and this article had just come out. And since then, these are a few of the articles that have come out in the Times about human evolution. So it seems like every week we learn something new. But it can be really confusing, because there's all these things that claim like, oh, it's the oldest, it sheds new light. And we need to somehow make sense of that. And that's what I'm going to try to do in this talk, is put all these different pieces together. But I want to start out. I'm a scientist. So I'm going to talk about evolution. And science has a limited scope that we can talk about. We can talk about things in the physical world, things that we can test. We can't talk about faith. We can't talk about religion. We can't talk about God. You can't test to see if there's a God. You can't test to see if faith exists or what it says. But I don't think they're completely incompatible. You can have both. I just want to get that on the table before we go, because I know human evolution, there's a lot of feelings around it. I want to make sure you know that. I don't think it's a struggle between faith and science. So if we stick with the science, I've seen this a lot, versions of this, right? Humans evolved from monkeys or apes or chimpanzees or what have you. Why do they still exist? When we think about this, we have to understand evolution, how it works, and it becomes really clear how they can still exist. So if we look, this is just a very simplified version of our primates. We've got our orangutans, our gorillas, our chimpanzees, our humans. No Bigfoot or Yeti, I'm disappointed. If we look down here, this is where chimpanzees and humans diverge. That means they shared a common ancestor. But that common ancestor wasn't a chimpanzee. Wasn't a human either. It was something entirely of its own. And once those lineages diverge, they start evolving into the forms that we see today. It's how we can have chimpanzees and humans existing at the same time. Another way you see this written is like, it's a straight line between monkeys and humans. That's really not how it works. We had some sort of common ancestor that didn't look exactly like a chimp, didn't look exactly like a human, and they diverged and gave us both. And if we look at our most distinctive human, I told you I need Bigfoot. The Patterson film. And if we look at some of the traits that we think are distinctively human, that separate our lineage from the chimpanzees and how we know which fossils belong in our lineage, bipedalism is a big one. Now the earliest of human fossils, potential human fossils, is this guy right here, Sahelanthropus chadensis. He gets the nickname Tumai. He's found in Chad, and in the local language, Tumai means something like child that brings forth plenty or rain. So they named it in hopes of finding more fossils just like it. It hasn't worked yet, but stay tuned. But all we have is this rather crushed skull. And I just told you the distinctive trait of humans are the hominid lineages that were bipedal. Well, how on earth can you tell if something's bipedal from the skull? You might think you'd need like the pelvis and the lower limbs and the things that we use for walking. But as it turns out, where your spinal cord enters the skull can tell us whether or not you were bipedal. If your spinal cord enters up underneath like ours, it makes your eyes face forward, which is pretty good evidence that you walk on 2 feet. If you're like a cat, your spinal cord is going to enter towards the back, and when you line it up, your eyes will face forward. Pretty good evidence that they're quadrupedal. And when we look at the reconstructed, that is not the real specimen. This is the man who found it. But using a cast, so he'd be murdered if he did this with the real specimen. You can see that it looks like it's up underneath, and Tumai would have been looking forward. Okay, and he comes from right about the time that we think humans and chimpanzees diverged, based on molecular evidence, so about 6 to 7 million years ago. However, like I showed you before, this skull is really, really crushed. So there's some arguments about how, when we try to put it back to the way it looked, where exactly that hole would be. So we still debate whether or not he was bipedal. If we want to get on a little bit firmer ground, we can go to Ardipithecus ramidus. He evolved around 4 million years ago. And unlike Tumai, I can say he, because we have a pelvis. I know that this one was male. And when we look at Artie, it's his nickname. We like nicknames. Okay, we can tell this is his skull. That spinal cord definitely entered up underneath. This is a reconstruction of Sahelanthropus for comparison. We're not quite sure. It looks like maybe an under a little bit towards the backside. This one looks distinctively human with that jaw jutting down, spinal cord going up. This is our first confirmed biped, 4.4 million years ago. But we've got a rather complete skeleton, which is great, although complicating things. It has this foot, and this is his big toe. And it does not look like our big toes, hopefully, if all's going well in your feet. Your big toe is lined up with the rest of your other toes. Artie's juts off to the side like a chimpanzee. So he was bipedal, but he wasn't bipedal in the same way that we are. And we have to go all the way forward until we get to Australopithecus afarensis, until we get something that looks bipedal the way that we today are bipedal. About the best known, best loved specimen of Australopithecus is this one right here, commonly known as Lucy. She was found by Donald Johanson in the 70s. The story goes that they were listening to Lucy in the Sky with Diamonds as they were cleaning the fossils back at camp, and that's how she got her nickname. There's an underground story that says that back at camp they all took LSD, and that's how she got her name. I don't know which is right. It wasn't around in the 70s. Now, up front, I do want to admit that this happened. Okay, and this is an important thing to mention in science. Sometimes we get things wrong. We just do. So someone, a graduate student actually, was examining casts of the Lucy skeleton. The real ones are very hard to get access to. You actually have to like FBI background checks. Was examining casts and said, I think the cervical vertebrae is not, that's one in your neck, is not a human. Okay, pretty sure it belongs to this guy, a baboon. Which isn't surprising. There were other animals around when Lucy was living. And fossils over millions of years do have a tendency to jumble themselves. It's not odd. But people made a huge, like, how did they not know about it? You know, what are the scientists doing? They're not good scientists. Well, the truth is that, you know, scientists are people. When they found Lucy, they were super excited about her pelvis and her femur because of all this bipedals and stuff. And they're super excited about her skull because we just all love skulls. And they just sort of put the other bones where they thought they went and kind of ignored them. They didn't pay close attention to any of the vertebrae because those weren't the really exciting, cool bits that you get published in Nature for. And nobody really took a close look at them until this graduate student did. But, to his credit, Donald Johanson admitted his mistake, because that's another important thing in science. We can get it wrong, we can have new evidence appear, and we have to admit it and move forward. Now, if I were to show you the difference between a baboon cervical vertebrae and a human cervical vertebrae, I would have a hard time picking them apart. So I also want to keep that in mind. This is very specialist and detailed knowledge. But moving on. So we have Lucy. We say, well, maybe there's problems. Let me go back a minute. Maybe there's some problems with Lucy's skeleton, like this part of the pelvis was deformed and had to be repositioned. And we only have one leg. Can we really say that she was bipedal? Well, the truth is Lucy is one almost complete specimen. This is when you know you're a paleontologist. You look at that and you're like, that's neat. Right. But we have over 300 elements from the same species. So there's one Lucy, it's true, but there are many, many other Australopithecus afarensis. And there's some whose pelvises aren't deformed like Lucy. And there's some who have femora that are more complete than Lucy. So we have a lot of evidence. We also have a set of trackways. Out in Tanzania, these are the Latolai trackways that were made by Australopithecus afarensis, so the same species as Lucy. And it shows a foot very much like our own foot. And these critters had the good fortune of walking across volcanic ash. It's like they knew geologists one day would want to date them. This volcanic ash is super easy to date using radiometric dating, so we can place it at the same time as the bones that we found. And all that adds up to Lucy and her kind being a biped. Now, sometimes we think about evolution like this. It's a very linear trajectory. We had our, not chimp, but our common ancestor, and then we sort of move forward through all the different taxa up till we get to modern humans, but that's not really how it was. So during the time of Lucy, there are many Australopithecines, all living at the same time. So many, many different species. And I don't expect you to be able to read this, but all the ones that are kind of in this orangey yellow, might even be tiger gold, these are all Australopithecines that were living round about the same time, which was right around 2 to 3 million years ago. And that's where we get this story that was published August 29th of this year, so a little over a week ago. And so we finally have a skull that says watershed in human ancestry. Well, it was the skull of another Australopithecine. This is Australopithecus anamensis, which was around at the same time as Lucy, Australopithecus afarensis. But we'd never had a skull before. That happens. Fossils, you get what you're given. Sometimes a bone fossilized, sometimes it's carried off by scavengers before it can fossilize. In this case, it was the first skull that we'd ever had of this species. Okay. I told you that a distinctive human trait, that how we can tell humans from the chimpanzee line, is that we became bipedal. But why? Right? Like, chimpanzee lineage didn't, and we did. Like, what's the difference? Well, it comes down to environment. So if we look at where these animals were living, in the Great Rift Valley, during the time that this split happened, we have the development of what every textbook will tell you is a mosaic environment. And what they mean by the word mosaic is that there's patches of forest and patches of grassland, and there's lakes, and there's rivers. And this is important for how we became bipedal. It's that there's patches. What that means is that to get the fruits and good eats that we like, we have to move in between the patches, which are not right next to one another. Okay. Oh, and another way that we know that it's this mosaic land is that we weren't the only things there. We get lots of other animals that we would expect to see in this sort of patchy woodland, patchy grassland area. We get hippos and lions and all sorts of ungulates. But what that patchy grassland did for us is it made us become really efficient walkers. In fact, if you look at chimpanzees walking, whether you look at them quadrupedally walking or you make them stand upright, bipedally walking, versus a human walking, you can measure all different, like the volume of air that they're expiring, calories that they're burning. But what the upshot of all of this data is, so I'm a scientist, I like to put up all the charts, is you get the cost of transport versus body mass, and you see that humans, we have a very low cost of transport. That means that we don't have to burn a lot of calories to walk a long ways. You can see our chimps up here, whether they're being quadrupedal or bipedal, whether those little dots are blue or red, expend a lot more energy to have to walk. So bipedalism is an adaptation to allow us to walk between these patches. But, okay, still, why didn't the chimps become bipedal? They must want tasty eats too, yeah? Well, all the early hominids are evolving right here in the Great Rift Valley, where we're getting these patchy woodlands because the plates are drifting apart. But the ancestor of human and chimps lived all over Africa. Africa's a big place. So there are parts, like over here, Congo, that aren't rifting, that maintain really dense wooded habitats. And the animals in those places evolved to become the chimpanzees. They didn't have to walk to get their goodies. They were living in the forest. And the animals that evolved to become humans live over here where they did have to walk. And I told you it was a patchy mosaic over here. Part of that is that there are big rivers. And even today, chimpanzees can't cross large rivers. So we have a barrier between these two populations. So they couldn't interbreed. So you have a barrier separating them, and you have different environments. And that's pretty much the textbook setup to get speciation, to get them to evolve into different species. So we think about our next very human trait. We like to think that we have big brains. I love the awkward Yeti. I teach anatomy, lots of good anatomy cartoons. But we really think that that's one of the things that distinguishes us from the rest of the animals. We're pretty smart. And in fact, when we look at the first fossils that we put in our own genus, Homo, they all have pretty big brains. But, well, are they just bigger animals? You know, whales have pretty big brains too, but we don't think that they're smarter than us. So one thing to do is to look at how the brain develops. And we're lucky enough to have fossils of juveniles of both Australopithecines. This is juvenile of the same species that Lucy belongs to. This is another Australopithecine, Africanus, lived down in South Africa. This is its discoverer, Raymond Dart. Raymond Dart is a character. If you ever get a chance to watch any of his interviews, do it, because they're fantastic. And over here, we have a juvenile. This is sometimes called, what's the old name for it? The new name is Narcotomi boy. The old name is Turkana boy. It just changed because of the people of the area call that area Narcotomi, not Turkana, so it's changing it from the name that white people gave it to the name that the people who live there have it. And if we look at how their brains develop, we see something really interesting. We see a difference between the way these Australopithecine brains develop and the way that the Homo brains develop. And you might ask yourself, well, how do we know they're juveniles? Maybe they're just small. That happens. We'll talk about habits later on. But one thing is we can see in the skull, these are all the adult teeth waiting to come in. Because we have two sets of teeth. We have baby or milk teeth, citrus teeth, and then we have the adult teeth. And you can see it in the fossils. And it turns out that chimpanzees, their sequence of eruptions is pretty similar to the human sequence of eruptions. So we figure we got it from the common ancestor. They probably have a similar sequence. And we can use that to help us estimate age. Another thing, if you ever get a chance to look at x-rays, radiographs of kids, like babies' skulls, they are frightening. All the teeth. Okay. So what we do is we look at the brains of the juvenile and compare their size, just the endocast, the size of the skull, to the adults. And we find, when we do that, in our more Australopithecine-like, this is actually a chimp, but Australopithecine-like, small brains, they reach full brain size by about age 3 or 4. And when we look, actually I have a better graph, yeah. So when we look at these early things, that's the tongue child, right around age 3 or 4, their brains are fully developed. And that's just slightly after chimpanzees reach full development. They reach it around 2 to 3. When we look at members of our own genus, Homo, when we look at Narcotomi boy, he is reaching final cranial capacity much, much later. Modern humans is around 20. If you teach undergrads, you may not think this is right. You may think it's slightly later, but developmentally, biologically, it's 20. Narcotomi boy falls somewhere in here, like 10 to 12. He's reaching full brain size, but it's taking a lot longer. And the longer that it takes you to develop your brain, the more things that you have to experience, the more things that you can learn. And in fact, when we look at the adult skulls, this is Australopithecine, we can see that in the Australopithecines, they don't have a forehead, the skull goes straight back, and it's sort of indented on either side, like a good place to pick it up. And when we look at the genus Homo, we see that we're starting to get a forehead and we're getting this expansion because our brains are getting bigger. And we start seeing things like stone tools. You don't see these beforehand. So brains are getting bigger. We're seeing evidence of some mental processes. Right? But why? Like, why on earth did we develop big brains? And there are lots of things, lots of stories in the news media. I don't want to quite call them hypotheses, but lots of stories about how humans got big brains. There's stories about how we've got these stone tools, maybe it's because we were eating meat and all the protein gave us big brains. My favorite is called the stoned ape hypothesis, where it's like we all of a sudden found psychedelics, and that somehow caused a change in our genetics and gave us big brains. Yeah, there's some wild theories out there. But if we go back to geology, to the plants, this is why plants are important, one of the many reasons, we noticed that the climate in the Great Rift Valley was changing. It was cycling between very, very dry and very, very wet during this time. And those cycles could take about 1,000 years, so pretty rapid cycling between wet jungle-like and dry savanna-like. And we know this in part because we can look at the geologic record, we can look at the rocks, and we see things like lakes that are big like the Great Lakes, forming, staying there for 800 years, and then completely disappearing, and then another 1,000 years later reforming. We can also look at sediments in the oceans. So we drill these ocean cores, and what happens in the ocean is all the dust from the continent sweeps off, gets deposited in the ocean, falls down to the ocean floor, and gets recorded in the record. So people who do this for a living, and I am not one of those people, can see the bands of really thick dust in the sediments and really thin dust in the sediments. You may think that's great. Okay, so we can see it was cycling, but how on earth do we know when that happened? Like, at least when we find lakes, we know that geologists can date rocks. Maybe we don't know how, but we know they can do it. But dust in the ocean? What? Well, it turns out there's these little critters that live in the ocean called diatoms. Microscopic, they have all these really pretty, they call them pests, but we can think of them as shells. And they're made out of calcium carbonate. Carbonate, which means they contain carbon. And they were living animals that at some point became non-living animals. So that means that we can carbon date them. And when we carbon date them, we find that these rapid cycles of having a lot of dust and then not a lot of dust were happening exactly when Homo was evolving in Africa. So it gives us another bit of evidence that we have this rapid cycling of environments. So that's really what those big brains are for. They're to help us to adapt to change. That's what differentiated us from all the other primate species that were living around at the time. We were able to adapt. So we're really not adapted for any particular climate. We're adapted for change. But then we have our next big event in human evolution. We had to move out of Africa. Okay, so if you look at a lot of the news stories, they're not about fossils in Africa. Anamensis was, but a lot of the newer ones aren't. It's like, what happened? How did we get out? Well, we did get out on foot. In the first wave, right after Homo habilis, we have Homo erectus, named because they are upright walkers. And those are the first critters that we see leaving Africa. Okay. And we see them leaving Africa around 2 million years ago. And it's not as if there was a band of them that got together and decided to be explorers and charged a course north. It's more like a little group here decided, oh, there's not a lot of resources. We got a lot of neighbors. Let's move up here where there's not as many. Okay, that's good. We'll stay here. And then maybe their kids are like, man, all these old people, we gotta move. And they moved up here, and you just get that sort of migration. So a slow migration. So it's going to take to get down here until about 800,000 years ago. Remember, I said they started at 2 million. So to get all the way over here takes until about 800,000 years ago. So they're moving slowly. This isn't a rapid migration. This is just moving to follow resources. These are Homo erectus. And then later, not everybody moved. I know which way my arrows work. Not everybody moved out of Africa. We still have some Homo erectus living in Africa during this time, just chilling. And they, due to other environmental pressures, start evolving and become modern Homo sapiens. Call them anatomically modern Homo sapiens. They look like us. And anatomically modern Homo sapiens They are going to migrate out of Africa, doing the same thing, following resources, not, exploring, trekking, around 125,000 years ago. So well after Homo erectus had already made its migration. Why am I telling you this? Well, it turns out one of our news stories, this was the first one, is about a discovery in a cave in the Philippines. And the critter that they found in there is a Homo erectus. You're like, okay, but you already told me they made it all the way to the Philippines. I saw the map. And I told you that they got there about 800,000 years ago. These bones date to somewhere between 67 and 80,000 years ago, which, if we are keeping score, is about the same time that anatomically modern Homo sapiens are in the area. So that means we have this, what we might call a relic population of Homo erectus living on this little island at the same time as anatomically modern Homo sapiens are in the area. They might have even met one another, traded with one another. We don't know. Now this was a huge story, right? Homo luzonensis. You get all these massive reconstructions of what they would have looked like, and they were rather short, diminutive. They get called hobbits sometimes. But if we look at the material that was actually found, we found some teeth, we found some toes, and we found some fragments of long bones. So, still early stages. So those reconstructions where they tell you exactly what the face looks like, don't buy them. They're based largely on Homo erectus species, specimens that we do know what the faces look like. But of course we know that these weren't the first hobbits to be found. We have Homo floresiensis, down here too in Indonesia. Also diminutive, sometimes gets called a hobbit. Also rather like a Homo erectus, living about the same times as the one on the islands in the Philippines. So more evidence that we had Homo erectus-like critters and anatomically modern Homo sapien critters living together. Right. So let's back it up. So I told you about the hobbits, and those make really big news stories, and everybody loves the hobbits. I do too. No shame. You might think, oh, but what about the Neanderthals? There is a lot of stories about the Neanderthals. Like, how do they fit into this picture? Well, this is our Homo erectus. Homo erectus migrating out of Africa, coming down here to be Java man and Peking man. Also coming to Europe. And the populations that live in Europe keep evolving, and that's where we get Neanderthals. We get these populations in Spain, going from Homo erectus to Homo heidelbergensis, and then to Homo neanderthalensis. So they're evolving from erectus species that lived here in Europe. Now Cro-Magnon, they are from anatomically modern Homo sapien species that moved out of Africa and evolved in the Middle East, and then kept moving towards moving along the Mediterranean coast. So Cro-Magnon and Neanderthals, not the same thing. Actually come from two different lineages. All right, so that's Neanderthals. But you may have heard there's this other group known as the Denisovans. And we've been finding a lot of stuff. They have been in the news quite often. You may think, well, who are they? Like, I've heard a lot of these names before, but not the Denisovans. So let's look at who they are. Oh, no, I lied. I have another guy to talk about before we hit, another news story to talk about before we hit the Denisovans. So back up. So you have Cro-Magnon. We've got anatomically modern Homo sapiens evolving, moving along the Mediterranean, these Cro-Magnon-like species. And the most conservative estimate up until June or July was that this happened, they started moving out around 125,000 years ago. They probably got up here maybe 100,000 years ago, maybe 80,000 years ago, somewhere in that range. Okay. But then we found this skull. hailed as the oldest modern human fossil, part of the anatomically modern Homo sapien lineage in Greece. And it dates to 200,000 years ago. So if we're keeping score, that is before I said they started migrating out of Africa. So that radically changes our views of what was happening with anatomically modern Homo sapiens. Now, it is rather new. Okay. And you can see this skull is in pretty bad shape. That happens with fossils, right? There's a lot of pressure that goes on in rock forming. It was chipped out of a limestone cave. And they did these CT scans, and they started reconstructing it on a computer without touching the actual fossil, which is very nice. But their dates haven't been verified. They've published it, it's past peer review, but we haven't had other geologists go in and date that area. Still waiting for those results. But if it's right, then anatomically modern Homo sapiens made it out of Africa way earlier than we expected. That's interesting, because this is the only fossil we have. And this is in Greece. And Greece is a long way from the Great Rift Valley. We don't have fossils along the way. And there could be some reasons for that aren't scientists are stupid or hiding things. There's some really good reasons. It could be that we just haven't been looking for them. You have to get money to mount expeditions to hunt for fossils. And we didn't expect to find anything older. So maybe a lot of people just thought, well, we've already found a lot of anatomically modern Homo sapiens from like 100,000 years ago. We don't need any more. We should put our resources elsewhere and not keep looking. It could be that the areas that they were staying in just don't produce fossils very well. It could be any number of reasons. But it is a good reason to mount further expeditions and look more. Now we have the Denisovans, like I promised. So a lot of news about this group called the Denisovans. We found jawbones. This was the first of the stories that entered the news. Jawbones in Tibet. They found partial skulls in northern China. There's been even more recent news about this group called the Denisovans. Right. So who are they and why do we care? Well, up until April this year, the only material we had of them came from this cave in Siberia. We had a toe bone, and we had a handful of teeth. But we had lots and lots of artifacts. And all the artifacts look very Neanderthal. And in fact, we could tell that they came from Europe because they were made of shells and other materials that we could trace back to European coastlines and areas. We didn't have much of the skeleton. Although teeth are awesome. If you ever study paleontology, you'll be welcome to the wonderful world of teeth. Teeth fossilize really well, and they protect that material that's inside of the tooth. The outside is made of enamel. It's almost a mineral on its own. It fossilizes like a champ and protects all the soft bits inside. And those soft bits got the geneticists excited because it contains DNA. They didn't live that long ago. They lived somewhere, well, this cave dates back to about 120,000 years ago. So the geneticists took material from the inside of the tooth and tried to figure out who they were based on their genetics. Because of course we have lots of Neanderthal genetics, and we have a whole bunch of modern Homo sapien genetics. So we can compare this new sample and see which one it's more like. Kind of like a very ancient paternity test. And what we found out was they looked like a mix. And that was exciting, but also didn't tell us much. In some ways, we're like, well, they're kind of like Neanderthals, and they're kind of like anatomically modern Homo sapiens, and we're not sure what's going on. There are all sorts of theories that perhaps this was a Homo erectus group that started interbreeding with Neanderthals, which would account for all the cool stuff that they had gotten from the Neanderthals, right? And that's why the genetics look like that. It's all sorts of theories, but kind of a side note, in most human evolution textbooks, usually used to show the power of genetics, or rather the limits of genetics and paleontology. But then we have all these news stories. And it turns out, so that's that cave way up here in Siberia, that a lot of the stuff that we've found is down here, more towards the southern part of the Indian subcontinent. And we've found skulls, and we've found, like I said, mandibles, jawbones, and it's accompanied by all the same cool artifacts, goods, that we had found up in this cave. So that extends the range of this group, the Denisovians. They're covering a large part of Asia. And if we looked at the genetics of the jaw found here. The jaw had teeth, right? Get the DNA out of it. It was up in Tibet. So we compared its genetics, both to Neanderthals and to anatomically modern Homo sapiens, and to different groups within anatomically modern Homo sapiens, or modern Homo sapien populations. And we found that they have the same gene that people living in Tibet have, that's associated with living at high altitude, adaptations and getting oxygen into your blood. Now, we're not sure if they got that from a common ancestor. If this population is maybe actually anatomically modern Homo sapien, these are ancestral to the people that live in Tibet today, or if they evolved it separately, because it's rather handy to have if you're going to live at high altitude. I haven't quite figured it out. It's only been, 4 or 5 months? Making progress. And we also started looking at, all right, well, we're not quite sure what they are. Some of the original thoughts were that they were Homo erectus, right? And then we start looking down here, like, well, if they moved here, and we have evidence that they were moving this way, maybe they are the ones that colonized these islands. And remember that the islands, that's where we found our little hobbits, Theresiensis and Luzonensis, and that they were, 67, 80,000 years ago, which is a large gap between, say, Java Man, the earliest Homo erectus that lived there, 800,000 years ago. So the thought now is that perhaps these really are a relict Homo erectus population, or the descendants of a Homo erectus population that moved down the Asian subcontinent and then colonized the islands. And that's why we're finding them there so recently. Okay, but you may still look and you may think, all right, well, the ones up here, we're not sure if they're anatomically modern Homo sapiens, Neanderthals, Homo erectus. That means they were probably pretty big, like our size. And you've told me that the things that live down here were really small. So why on earth would you think that these guys would be the ancestors of these guys? That doesn't make sense. Well, it turns out that when you live on an island, weird things happen. We know this in a lot of other animals, not just primates or Homo sapiens. And it has to do with resources. If you're on a small island, not Australia, this is too big. But these little islands, small enough, you have limited resources. So if you're an herbivore, you're a frugivore, you're not gonna make it if you're huge. Like an elephant does not make it on an island, right? They just need too much food. So you see selection for smaller body forms. And I mention elephants because we have dwarf mammoths off the coast of California. Many other dwarf things, dwarf camelids. Lots of animals that we think of as being very large herbivores dwarf on islands. So it makes sense. We're kind of a big herbivore, frugivore, omnivore, that we would also reduce in body size. And you may think, well, but wait. I know that Komodo dragons are an island species too. And they got big on islands. Like, what's up with that? Well, it sort of depends on what you eat. I was talking about mammoths. They eat plants. Komodo dragons, they eat other animals. If you're on an island, usually the top carnivores have been knocked out. They just can't make it. Like your lions and your tigers, there's just not enough food for them. What you start seeing is sort of small and medium-sized carnivores arrive on islands, especially reptiles that can eat one meal and be fine for months. And they start growing larger because now there's nothing else eating all the little dwarf mammoths or the little hobbits or whatever our little herbivores are. So both things can happen on islands. And with that, I wanted to make sure to tell you guys that I am teaching a class on human evolution this spring. So Biology 470, it's a special topics class, Monday, Wednesday, Friday, hour-long lectures, in case you want to know more. And if you want to know more right now, I made sure to leave some time. It's supposed to be a 50-minute talk, so I think we got like 7 minutes, but I'll stay here as long as you want to ask me questions about human evolution. Or anything. Yes?
So, in getting into the islands off of Australia, they have more talking technology back in the they have boats, they make boats.
But if we look at the islands, today there's big gaps between them. But sea level was different when these things were moving around. Sea level was much lower, right? So you've got a lot of water being pulled up into the ice in the polar caps. So first off is they're much closer together. But they still would have needed to cross water. They could have made it in very primitive rafting-like situations. So if we even think about like how monkeys got to the New World, like vegetation rafts, not rafts that you carefully create and think you're going to set out on a trip on, but things that just sort of like naturally occur during a storm and wash you away. That's how we're thinking that they got there. So maybe not super on purpose.
Okay.
So do they know how evolution actually happens? Like I read that it has to happen in the sexual organ of the body, so they push the new gene. Like how does evolution affect like your sexual genes?
It's a good question. And let me figure out where to start. So for evolution to happen, we do have to have the trait, needs to be heritable, which is why it has to happen in the genes in your egg or your sperm, if you're a vertebrate, or if you're a mammal. Birds are weird.
They have it, but they're sex chromosomes.
XWs and
Yeah.
Anyway, so it has to happen in those sex chromosomes. So it's not as if the environment is affecting your eggs or your sperm. What's happening is that there's variation in traits. We look around, everybody looks different. Some of us are taller, some of us are shorter, some of us maybe have better eyesight than others. There's just natural variation in a population. And what happens with evolution is that the environment, some of those variations are going to be more successful. Maybe you're in an environment where being tall, you're going to be able to get more food. And if you can get more food, you can have more babies, which means more of your babies are going to have that tall gene. And the people who are shorter, they don't get as much food. They don't have as many babies. So there are more tall genes in the next generation than there are short. And that's going to keep happening as long as the environment's the same. People with the tall genes are going to keep having more babies, and it's going to spread throughout the population. And that's the same thing that's happening here. So with bipedalism, you had animals that had hips that were shaped just slightly differently. Just variation, our hips are all shaped slightly differently too. But some of these differences gave those animals an advantage. Advantage in getting food or in getting mates, some sort of advantage that let them have more babies. So that's what evolution cares about, is how many babies you have. So those babies had the same hips, and they create more babies that have all those same hips.
Yeah, what I'm confused about though is the variation. So like, if the variation is different for each human, how does it select those characteristics? Like, so like, if humans if everything that gets born pretty much is very different, how does it select those certain characteristics when you have another child? So like, and the child's gonna be very varied from you too.
That's right. So there's just a greater chance that it's going to have those genes. It's not 100%, but if you have a lot of babies, more of them are going to have that same gene than not.
Oh, that makes sense.
Yeah. There's a lot of chance involved in evolution. That's why statistics is important if you want to go into science.
I love that you just said that. So have you seen, I believe, there was an article that came out, it was June or July, the book came out before that, Dr. Stephen Meyer's Darwin's Doubt. So he has an argument in there where it's actually, and he takes a bunch of studies from Axe and some previous biologists, where it's mathematically impossible to select new body forms via any neo-Darwinist sort of paradigm in biology. And he uses the Cambrian explosion, but it works fine for this when you're talking about selecting for an upright torso or a different skull. It was the chance of finding a actual working protein was 1 in 10 to the 77th power, and that's over the course of from the beginning of Earth 14 billion years ago, and obviously we don't even have that long for life if you're talking about when actual multicellular organisms were to come about.
4.5.
And so, with that information, how exactly do we rationalize this sort of generally slow, when you think about the age of life on Earth, or actually fast, sort of progression from our common ancestors with chimpanzees who were quadruped to these sort of beings right now walking on 2 legs, skull placement's different, obviously hips are different, musculature when you talk about the skulls and the muscles going up to the bridge of the cranium. I mean, how does all of that change if it's mathematically impossible to make even new singular proteins, let alone protein folds, to sort of dictate those body plans?
Okay, so this is a very big question. I'm going to try to answer it in a few parts, but I'm not going to be able to completely answer it right now. I think that's almost a whole course. One, when you say it's mathematically impossible, mathematicians and physicists sat down and figured out that bumblebee flight is mathematically impossible. I've seen bumblebees. They fly. So that means there's something wrong in the reasoning there. Something's gone wrong in the math and the physics. And I don't know what that is. But at least in that case, I can look and I can say, like, it has to happen. Because look, right there, it's happening. And this is what I'm guessing might be the case with this study. Because I haven't looked at it, and I'm not a statistician. But my guess is that that study's based on making certain assumptions about the way the natural world works. And that maybe some of those assumptions are faulty in a particular way, and that's why we're getting the result that we are. Because we have seen proteins change. Even in modern animals, if we look at dog breeding, there's still work going on the Galapagos finches, if you would believe that. And a new species of finch was recently discovered. And we can trace the changes at a molecular biochemical level in those organisms because they're all still around. My guess is that there's some assumption.
So the dogs and finches, though, aren't body plan changes. What I'm talking about is body plan changes.
Well, a
little bit bigger of a beak, that's like saying there's a body plan change between you and I just because one of us is taller than the other. And that's not necessarily the case.
Let's see.
Well,
So-
the beaks did change features.
What?
The beaks of the finches, they did change their beaks. So that's the body change.
Yeah.
And he does argue that species differentiation between like dogs or finches, for instance, that's different than changing from, say, a quadruped to a biped, or changing from, say, like a fish into a lizard. And these changes take too long to be explained by any sort of neo-Darwinist theory.
I know the study you're talking
Oh,
about.
good.
When you're talking about this massively improbable protein pool, you're talking about going from de novo with no selection. I mean, there are a huge amount of card hands you can deal with, and they're all equally improbable. But what's happening in this case is that the selection is allowing certain things to happen. So that actually speeds up the process.
But when we're talking about selecting for genes inside the egg or the sperm, it has nothing to base that off of. So I can select how my body needs to improve because I can interact with the environment. My sperm cannot do that.
Right, but
And so
the
when-
offspring that are going to be more fit in whatever that environment are.
But then what's the mechanism that chooses what is fit at that point? That's what I'm saying. Your
It's-
DNA doesn't just change.
Yes,
Yes, it does.
Yes,
Okay,
yes.
there's mutation, but
Yep.
the amount, the chance that it is a functional protein or because even a singular protein is the one in 1077. A protein full would require far more data.
Right, but if
And
you've
the
got,
ti- and
if
the-
you've got
the-
10 to the 77 trials out here, and you put those in an environment where one is more able to survive in that, then that's the one that's gonna become more dominant. That's just like, we could deal out poker hands to everybody on campus, and the chances of us having the same hand are gonna be massively improbable. But if something selects for that one to be more able to survive in that environment, then that's the one that's gonna become more dominant.
That is to say that there was 10 to the 77th power of whatever the common ancestor was, and they had one that was
Yeah,
capable of
but you've
taking
got
that off.
3 billion base pairs in the human genome. There's a lot of shuffling that goes on in
And
mitosis.
a change of a single one of those sequences could ruin the organism.
Yep.
Exactly.
And so you have more of a chance of getting a faulty protein. So we're set, I mean, 10 to the 77th, I can't even pick out exactly how large that number is, but I'm not a math person. But you're saying that there was 10 to the 77th of whatever the common ancestors between chimpanzees and human beings were. And one of them made Homo erectus. Now we have one Homo erectus.
We're skipping many steps.
Yeah, it's not that you're talking about the tornado in the junkyard fallacy, where a tornado sweeps through a junkyard and the chances of assembling a 747 are impossible. But evolution doesn't work that way. You start off with simpler forms, and those are selected by the environment, and then those become more able to survive in that environment, and they have more babies that are able to survive. So giving an incredible amount of time and an incredible number of genes that can shuffle, it's well within. I mean, look at antibiotic resistance. I discovered antibiotics, penicillin in the 40s. Now it hardly works anymore. Because there's so many organisms and so many changes, possibilities.
Right, and if we think about things like Hox genes are master control genes, so a change in one of them can have many, many downstream effects.
But then a Hox gene would require the functioning proteins, to which I already said the ratio of failed to successful proteins
Right.
is infinitesimal.
But when proteins are folding inside the cell, they're not folding randomly. And then I think that's one of the common misunderstandings, is that this is all random, and it's not. There are other proteins that are helping those proteins fold into the correct shape, and the ones that survive that correct shape are the ones that leave a viable cell. But you're right, if you came in and just said, okay, click and scramble all proteins and create eggs out of all of those,
That's where the math fallacy is. Not
It's the-
all
it's the
the
selection,
forms
right.
are biologically possible. That's why you get such a large number. I think there was some assumption hidden in there. I did just want to point out, like, so we talk about these differences in shape. If I put a chimpanzee pelvis and a human pelvis, and Australopithecus pelvis, like models of them, a lot of people, including law enforcement officers, can't tell them apart. They look super similar. So it's a very small change in the way these things are oriented that creates bipedalism too. So you have a small change that's creating a big downstream effect.
So I want to add something. Also, one gene can change it where you have hair on your entire body versus not.
Yep,
So,
hirsutism.
yeah. So it's like it's not likely that it is happening by random. It's just I think the DNA just keeps track of everything that's ever been in the organism, and those genes get turned off or the other ones are turned on, right?
Pretty much, we call that epigenetics. So there's these little methyl groups that attach to the DNA and sort of tell it where to start, like transcribe this protein, but not this protein. So you're right, sometimes it just turns off a protein and you get a change in an organism, and sometimes it turns on the creation of a protein and you get that change that you can see in the organism. But then sometimes throughout evolution you also get like changes in proteins that weren't being coded for. And because it doesn't affect the organism, nothing repairs it. So eventually those become useless. other questions? I'll also look at this side of the room, since I've been facing this way. All right. See where we are? Oh, and we are right on time. So, I'll stick around, but I'll release you into the wild. Go forth.